Couple keystone statements to get us started today. These two talk about why DNA is important. Here's some more about cells growing and needing to divide, which is what we're going to talk about today, cell division. It's also called the cell cycle. Happens in eukaryotic cells, what we're going to talk about today. And in them, they actually divide their nucleus first and then the cytoplasm to make two new cells. You'll probably remember this from bio. It's called mitosis. The word mitos in Greek means thread or yarn. And if you look at those pictures, it looks like some piles of yarn in the middle of those cells. That's why it got the name. Under the microscope, it looks like piles of yarn, some yarn process or thready process. The word mitosis actually has nothing to do with what's happening and everything to do with what it looks like. This happens in your normal cells, which are for just everyday functioning. It produces clones of those cells. The first cell, before it divides, is called a mother, and the two cells that are produced are called daughter cells. And they have exactly the same DNA. The DNA is identical in them. So we divide this into four components, sections, or phases. It's kind of like growing up. You're not a child one day and an adult the next. So we don't go from one phase to the next in this process in steps, but it's a continuous process. We just give it names so we can say, oh, this is what's going on there. So at the beginning of a book, you might find a prologue, because pro means first or before. And so prophase is the first phase where the cell actually begins to prepare for this process. I like to compare this to moving from one house to another. When you move, you have to pack all of your things into boxes. You can't use them. Life is not easy or convenient. And the same way, all normal life for the cell stops during mitosis. All the DNA is packed away. There's little gummy worm looking things in there are the chromosomes. That's the condensed DNA that's packed up in boxes, ready to divide it into two parts. What you're actually looking at there is an egg from a fish with one cell inside, and then the nucleus has the chromosomes in it. So that material that the chromosomes are made of is called chromatin. It's the DNA with the supporting proteins. The nuclear membrane dissolves so that we can organize the chromosomes. Each chromosome at this point is X-shaped. It has two parts called chromatids. These were made through DNA replication. So they're identical copies. So the dark purple and the light purple, identical copies. Whatever's on one side in one is on the other side in the other. They're held together by a structure called a centromere. The kinetochore actually is where microtubules attach to pull them apart. And so DNA replication happens so that there are these two identical sisters that can be separated in mitosis. Without DNA replication, cells couldn't divide because cells wouldn't have their own copy of DNA. Prefix meta means middle. And so in metaphase, the chromosomes line up along the equator in the middle, getting ready for the two halves to split. And so the nuclear membrane's gone, microtubules attach. We call this a spindle because of a structure on a spinning wheel to make yarn. We don't recognize that structure anymore, but it still has the same name. It looks like that. So that's called a spindle. It's attached to the centrioles, which are going to pull to opposite ends. And so when they actually start backing away from each other, the prefix ana means back. If you remember in metabolism in the last unit, anabolism is the putting back of the carbons into complex molecules. And so ana means back or backing up. And so the chromatids back away from each other. The X's come apart, and we start to see the division of the nucleus. The prefix telo in Greek, tele in Latin, means far away, and so telophase is the far away phase where the chromosomes get as far away as they can. Then this process of cytokinesis begins. This is late telophase, where the cytoplasm actually divides. That word in three parts would literally mean the process of cells moving. The events of prophase are reversed, which means the chromatin gets unpacked out of the boxes. The nuclear membrane comes back. It's time for life to start again. The daughter cells are identical. They have each exactly the same DNA in them. And here you see the classic figure 8 pattern that telophase shows. That again, there's the outside of the egg. Now there's two cells, almost. They're pinching off. They're being cut. The word cleave means to cut, and so that's called a cleavage furrow or cleavage ditch, cutting ditch. And so the cytoplasm is dividing there, separating into two cells. And then we have this other part that we talk about with mitosis, and it's very unfortunate. It's called interphase, which would literally mean the in-between phase or the not mitosis phase. This is actually the most important part of a cell's life. This is when all normal cell functioning happens. It's really, really important, and so it's very unfortunate it has this name. And so you see the chromatin there fills up the whole nucleus. It's all spread out. It's unpacked out of its boxes so that the cell can use it to do the things that we're going to talk about later, which is what the DNA is actually used for. And so here's a